CCM nerve image analysis. In this video we're going to discuss how to analyze the corneal nerve morphology using CC metrics. CCM nerve image analysis involves three steps. Step 1. Trace the main nerve fibers. A fiber longer than 50% of the image length without its endings branching to another fiber is a main fiber. Step 2. Trace the branches with a blue line. A fiber arising from a main fiber is a branch. And step 3. Mark the branches on the main fibers on the red lines with a green dot. To analyze the corneal nerve morphology using CC metrics, click on the right top corner DC and NFD to trace the main nerve fibers with a red line, and then to trace the branches, click on NFL to trace them with a blue line. You can mark the branches with a green dot by clicking on NBD. There are 10 rules to analyze the corneal nerve fiber morphology. Rule 1. Trace the fibers from left to right in the image. Rule 2. A main nerve fiber is longer than 50% of the image length and its two endings do not branch to another fiber, whereas a branch is shorter than 50% of the image length, or even if longer than 50% of the image length, as long as it is connected to a main fiber. Rule 3. Regardless of the fiber's width, orientation, including linear, curvy or tortuous, direction or transparency, meaning out of focus fibers, if it is longer than 50% of the image length, does not cross with another fiber, and its two endings do not branch to another fiber, it is a main fiber. Rule 4. Mark the branches with a green dot on the main fibers, the red lines only. Do not mark the sub-branches, the blue lines. Rule 5. If two fibers intersect, one of them is the main fiber and the other is the branch. The main fiber is either the widest one, if the width is different between the two fibers, or it has the same orientation or direction with the original fiber before the split. So you see, this is before the split, and that has the same orientation, and that's why we trace it as a red, whereas the other one is the branch. Here's another example where this is before the split, and then that continues in the same orientation and direction. That's why we trace it as red. Similarly here, this one we trace it red because that one has the same direction orientation. Rule 6. Two fibers crossing each other are main fibers if they have similar width and the endings do not branch to another fiber. So you can see these two has the same width, and that's why we trace them as two main fibers. Here's another example that we apply Rule 6 with a different morphology where there's a longer overlap between the two main fibers. We still trace them as two main fibers because the width is similar. Rule 7. If two fibers of different width cross each other, the main fiber is the wider fiber and the branch are the thinner fibers. So looking at this example here, this is wider and that is thinner, so this would be the branches. Instead of having two main fibers, we have one main fiber and two branches. Rule 8. If two fibers cross each other and the endings do not branch to another fiber, but it is not possible to determine the width of the fibers due to the short length of one of the fibers after the split, the fiber with similar orientation direction or transparency before and after the split is the main fiber, and the other fiber is the branch. Rule 9. If a fiber is vague but distinguishable and doesn't require guessing, it should be traced. If it is vague and requires guessing, it should not be traced. So looking at this example, these fibers here are distinguishable, and that's why we trace them. Here's another example. Right here, that's a pressure line. So the fiber is a bit vague, but it's still distinguishable, and that's why we trace it. Here, the fibers are not very clear. There are dots and there are quite a few gaps. It requires guessing, otherwise if we trace this, we may end up tracing a lot more things that are not supposed to be traced. So we haven't traced this. Rule number 10. If the width of a short fiber is homogeneous, it is a branch. A Langerhans cell is short and its width is heterogeneous. Langerhans cell should not be traced unless it is attached along a nerve fiber. So looking here in our example, that is a Langerhans cell. It comes in different shapes and forms. Some of them are short, some of them are long, and they look like branches. But the width is heterogeneous, it keeps changing from thin to thick. Here's an example of another Langerhans cell that looks like a branch. We will not trace this. As you can see, its width varies. This is short. However, this Langerhans cell is attached to a fiber. And that you can tell it from because it extends beyond the Langerhans cell, and that looks like a fiber that rests. So that is why we would trace this, even though the Langerhans cell is attached to it. So I hope you found this video useful, and if you did, click on the like button, subscribe to our channels for future videos, and thank you for watching.